Hello everyone, welcome to MMBS111 Introductory Microbiology. This is mini lecture 13.5. We're talking about stages 3 and 4 again, but this time we're going to be discussing the B cell response, how B cells are activated, and their role in acquired immunity. There are 5 learning outcomes for this mini lecture. First, know the 6 steps of B cell activation and be able to identify the players. Describe the structure of an antibody and what is the purpose of the variable regions and the hypervariable regions. Know the different antibody functions. Name the classes of immunoglobulin and function of each. And finally, what's the difference between primary and secondary response to antigens? So we're going to start by looking at this diagram from your text. It can be found on page 364. And this illustrates B cell activation. So at the same time that T cells are being activated, we also have B cells that are being activated. And there are a number of steps in this process. So in order for the B cell to be activated, we first need to have a reaction with an antigen. So the B cell is going to recognize that specific epitope of its single specific antigen that it recognizes. Excuse me. Once that B cell has bound to that antigen, then there's going to be some processing of that antigen within the B cell, and then it's going to present that antigen to a T helper cell using that MHC2 marker on its cell membrane. And once we have that recognition, or that presentation of the antigen by the MHC2 marker, we're also going to have some recognition by a T helper cell. This T helper cell has to be already activated by an antigen presenting cell. So an antigen presenting cell like our dendrites are going to activate that T helper cell, as we saw in the previous section. That T helper cell is going to recognize that presented antigen on the B cell, and then we're going to have some interleukins and some cytokines that are going to be released that are going to stimulate B cell activation. Once the B cell has been activated by the T helper cell, then that B cell can begin to differentiate. So we're going to have proliferation of the plasma cells that are going to produce our antibodies. We're going to have the production and proliferation of memory cells that will remember that antigen. So when it is exposed to that antigen a second or third or fourth time, that response can occur much more quickly because we have B cells that remember that antigen. And we're also going to have the production of regulatory B cells that are going to help proliferate more B cells, and also they help to regulate the T cell response. So these are the steps involved in B cell activation. Make sure that you understand what's happening here and the the cells and structures that are involved in this process. So our B cells are the cells that are responsible for antibody production. You can see on the image here, this comes from your text. This is a diagram of an antibody. And it is pretty standard. We'll take a look at different types of immunoglobulins, but essentially these are the components. So the antibody has typically on the simple Y-shaped form like IgG, it has two binding sites, one on each end of the Y. It's composed of two long, heavy chains that are bound together with disulfide bonds, and then it has two light or short chains that are also bound to the heavy chains with disulfide bonds. The top section is the variable region, and you can see it's marked there with a V, and it's colored yellow. That's the hypervariable region. This is the region that we discussed earlier that is coded for with 500 different gene segments, and that leads to the variability that is present in this compound. Those antigen binding sites are going to be very specific to that single antigen that antibody recognizes. You can also see at the end of the heavy chain what's labeled the binding site for cells. That is the location or the site that the antibody is going to bind to the B cell on its cell membrane. The Fab region is the variable region that contains the antigen binding sites. We're going to talk a little bit more about the FC region a little bit later in this presentation. So antibodies can interact with antigens in a number of different ways. That hypervariable region, as we mentioned earlier, is the site on the antibody where the epitope binds. And as we've mentioned before, that region is very specific to a single epitope. The amino acid content of that hypervariable region is extremely varied. As we mentioned before, that's what contributes to the multitude of varieties of antigens that can be recognized by antibodies. The specificity between the antigen binding site and the antigen or the epitope is very similar to the way enzymes recognize a substrate. It works similar to that lock and key mechanism, so the antigen is very specific and it must fit the site on that antibody. And each of those antigen binding sites on a single antibody is identical. So those two Fab sites, one on each end, are identical to each other. They can bind an epitope on the same cell or on different cells, which is going to contribute to the variety of ways that antibodies can interact with the antigen. So table 13.7 comes from your text. You can find it on page 365. This illustrates the number of ways that antibodies can interact with antigens. So you can see our first example that antibodies are able to coat the surface of a bacterium by recognizing certain proteins or markers on the surface of those cells. They can also interact through a method called opsonization, and this is a process that makes the microorganism more recognizable by phagocytic cells. So this sort of coding process that you've seen here in the first image actually makes the phagocytes more aware of those microbes and better able to phagocytize them. Another way that antibodies can interact with antigens is through something called neutralization. And this typically happens when we're talking about a virus. So the antibodies can bind to certain receptors and sites on the virus, and in that binding, they inactivate those sites on the virus to prevent them from attaching as they normally would to the receptors on the cells of tissues that they naturally recognize. And that is one way that our immune system is able to prevent that adhesion process that viruses use in order to enter cells. Another way that antibodies interact with antigens is known as agglutination. And this reaction is used in a number of immune tests, including the test to determine blood type. So we actually look for agglutination with certain antibodies to determine blood type. Because antibodies can have two identical antigen binding sites on each end, they're able to bind more than one antigen at the same time. So this can result in a clumping or aggregating reaction that's known as agglutination. And this prevents the microbes from moving around. It also makes them more noticeable by phagocytes so that they can be removed from the body. Another way that antibodies interact with antigen is in conjunction with complement, and that can result in lysing of bacterial cells. And then finally, antibodies can also interact with toxins, and we call these antitoxins, and they are able to recognize and neutralize exotoxins produced by microorganisms. All right, so let's talk briefly about the function of the Fc part of the antibody. So this section of the antibody contains an effector portion that can bind to receptors on the membranes of cells like macrophages. And the effect of the binding of that section to a cell is determined by the cell's role. And this can be involved in opsonization, where the antibody is going to attach to foreign cells, and it's also followed by the binding of that Fc portion to the membrane of the phagocytes. And then that allows the phagocytes to be more effective, and they're able to engulf that opsonized microorganism. These are the 5 different types of immunoglobulins or antibodies. You can see that each class is identified on the far left. And then you can see there in the next column the structure of each. So most antibodies are monomers. They're composed of a single Y-shaped structure. However, there are a couple that are composed of two or more of those Y-shaped subunits. You can also see here the percent of the total serum. So how long each of one of these antibodies generally lasts in the serum, and then what percentage of antibody production is composed of each of these antibodies. And then you can see the properties of each as well as the functions. So IgG is the most common antibody. It is a monomer, so it's composed of a single Y unit. It lasts about 21 days in the serum, and it makes up 80 to 85% of the antibody that is found in blood serum. It has a number of different properties and functions. So it specifically attaches to phagocytes. It's also involved in complement fixation. It is I think the only actually antibody that is able to cross the placenta. So IgG results in temporary immunity in the newborn. It functions in agglutination, opsonization, and complement activation. IgM is the next most common antibody. It is a pentamer, so it's composed of 5 different Y subunits. Lasts about 10 days in the serum, and it composes about 5 to 13% of antibody production. It's involved in complement fixation. It's the first antibody that is produced during the primary immune response. And it is the only class of antibody that is produced in response to T-independent antigens. It is involved in agglutination and also complement activation. IgA is the next most common antibody. It can be found in 2 forms, as a monomer or as a dimer, and it is a secretory antibody, so it's secreted by B cells. Lasts about 6 days in the serum. It is secreted in saliva, breast milk. It's found in the mucus and mucous membranes. It also, the secretory form is also able to resist enzymatic degradation. And this monomer is primarily responsible for protection in the mucous membranes. So it prevents attachment of microorganisms in things like the mucosa lining of the alimentary canal, the lungs, trachea, anywhere that we have mucus secretions in a mucous membrane. IgD is the next most common antibody. It is also a monomer. It makes up less than 1% of antibody production, and it lasts about 3 days. It's involved in the development and maturation of the antibody response. It's not clearly understood all of the roles and locations of IgD. The final antibody is IgE. It is also a monomer. Lasts about 2 days in the serum, and it makes up about less than 0.01% of antibody production. It is mainly involved in allergic reactions, and it also helps to attach to and expel parasites. Its Fc region is going to attach to mast cells and basophils. And it also is going to stimulate the release of the granule contents of some of the granulocytes, like eosinophils and basophils. So those are our antibody types. Make sure that you know these and you understand their functions and where they are going to primarily be found. This graph is also from the text, and this illustrates the primary and secondary response to antigens and antibody production. So you can see in the first section is the primary response. So this is the first exposure to the antigen. And in the first exposure, there is this latent period where antibodies are not being produced, but B lymphocytes have been activated, and this is where we're seeing the proliferation of clones that recognize that particular antigen. We're going to eventually have plasma cells that are going to produce antibodies, but it takes time for the B lymphocytes to first become activated and then produce clones that will recognize that antigen. Most of these antibodies are going to be IgM type. These are the first class of antibodies that are secreted by plasma cells, and then later on we will have more IgG produced. So after that initial response and we have that antibody production and the antigen is then removed, then we have this space of time after the initial response where there's no activity. During this time, we're going to have memory cells that were produced during the primary response that are going to continue to circulate through the body, and they will be the cells that are going to respond in the secondary response when we have when that antigen is experienced for a second time. So you can see that there is no latent period. Antibody production is almost immediate once the antigen is recognized. You can also see that we have more IgG produced overall in the secondary response, and the antibody titer is going to be significantly more in the secondary response than in the primary response. And this really has an advantage to us because this allows for a much more rapid response to an infection with the inclusion and the maintenance of memory cells that are able to recognize these antigens immediately and then begin producing antibody immediately. That is the end of section 13.5. These are some checking in questions. Make sure you go through these and you can answer them correctly. Also make sure that you go through the learning outcomes. Make sure that you understand those well. You feel like you've mastered them. Here's our critical thinking question. Respiratory syncytial virus, or RSV, is a viral infection of the respiratory mucosa, more predominant in young children. Which antibody would be responsible for the neutralization of this virus? So think about our antibodies, where they're located and what they do, and formulate your response, and then you can compare how you would respond to my response on the next slide, and hopefully this video was helpful.